I have the privilege of starting out our scientific program, and our first speaker is Leah Addadi. So Leah is one of those scientists that has been working on nanotechnology for a long time without even knowing it. So this speaks to her vision. So it's been decades that she has been exploring all the nanoscale complexities of how minerals form in biological systems. And today, her research touches on many areas. It's relevant to pathological conditions in humans in the context of mineralization, osteoarthritis, osteoporosis, and she even thinks about amyloid formation in Alzheimer's disease, many interesting problems, but also her research, I think, can open up many biomimetic approaches to nanoscale designed devices in the future. So her research really covers a very wide range of subjects. So Leah is originally from Italy. She studied in Padova. And so if you're wondering why she has an Italian accent, or sort of, then you'll know why. And then she immigrated to Israel after studying chemistry in Padova. She went to the Weizmann Institute in Rehovot and got a PhD in structural chemistry. She worked with a famous crystallographer, Meir Lahav. And she, after finishing her PhD, she went to Harvard University to do her postdoc with Jeremy Knowles. And eventually she ended up back at the Weizmann Institute, where now she is the Patrick Gorman Chair of Structural Biology at the Weizmann Institute. So Leah has been recognized by many interesting awards and honors. I will just mention one because it's very timely, perhaps. Leah will receive in 2011, coming up, the Aminov Prize, which is given, is the Aminov Prize for crystallography, given by the Royal Swedish Academy of Sciences. So congratulations, Leah, for this recent award. She is going to delight us today with a story. She won't be able to talk about everything she does, but she's going to tell us about guanine-based biogenic photonic crystal arrays in fish and spiders. Please help me welcome my friend and colleague, Leah Addadi.
Well, good morning, everybody, and thank you, Sam, for this wonderful introduction. Actually, you've made things very easy for me now, to start at least. I'm very excited to be here and very honored to have been given the task to open this day. Although, as Sam mentioned, I'm in fact not you won't hear from me the common, the things that you're used to hear about nanotechnology, insofar as we are not building anything. We are rather trying to understand what organisms do in biomineralization during the processes through which they build their skeletal materials and also many other solid materials that are used for different purposes. And in fact, it's been now many years that together with my colleague and collaborator, Steve Weiner, we've been looking at these processes. And the surprise, or maybe not a surprise, is that this is all about nanotechnology, as Sam said. Because organisms have had more than hundreds of millions of years to optimize the strategies and the design principles of these mineralized materials. And they do that by exercising control from the molecular scale, from the nanoscale, up to the centimeter scale. So they are a very good place to get inspiration for nanotechnology. And just to give you one example of things that we've been doing, but that I won't talk about, if you look at these fantastic skeletal elements, a vertebra of a brittle star, these spicules of sponges, the spicules of the embryos of sea urchin, and the big, mature sea urchin spine, it comes as a surprise to realize that these are, in fact, single crystals from the crystallographic point of view. They are single crystals of calcium carbonate, of calcite. And then, obviously, the first thing that comes to mind is how can they do that? How can they build a single crystal with such a convoluted and complicated morphologies that is faithfully repeated by all the organisms during their development? But today, I want to talk about a different phenomenon that is not so well known and not so well studied. And these are the optical manipulation of biominerals. There are not many cases that are known of that. One case that became quite well known about 10 years ago is the case of the brittle stars. There are some brittle stars that are sensitive to light, and it turns out that what they have developed on the upper part of their arms are these fan-like elements that have these bumps that are in fact microlenses. They have a perfect geometry of an optical lens, and they concentrate light onto a focal point in a position where the nervous system of the brittle star resides. Now, interestingly, the brittle stars are closely related to the sea urchins that I was showing you before, so that most probably the mechanism whereby these microlenses are produced and these elements are produced is in fact the same as I was showing you before for the sea urchin spicules and spine. Another example that has been studied by Jan Eisenberg a few years ago is sponge spicules that have the structure of real light guides. Although these are made of silica, that's glass. Although it is not clear that the function is in fact a function of light guides, but it's striking as they are built, how they are built, and they can in fact function as light guides. Again, what I'm going to talk today is something slightly different, and these are structural colors. Structural colors are these beautiful colors that we are used to see in quite a number of organisms, and it is impossible to look at how beautiful they are without asking ourselves how they are made. And in fact, a long, long time ago, in 1665, Robert Hooke is already asked himself how these colors can be produced and tried to explain it for peacock feathers. Doesn't matter that he didn't get it right at the time, but we progressed since then a little bit. They have evolved over 500 million years ago, and they are quite widespread. So there are examples in insects, in arachnoids, in nematodes, in birds. And some of them, such as in fish, that we'll talk about quite a bit, in beetles, in spiders, in butterflies, they involve highly reflective surfaces. And the phenomenon is based on thin film interference that may be single layer or multiple layer. In single layers, thin film interference is caused by the reflection from two interfaces that delimit the thin film in contact with a medium of a different index of refraction. And this is the case, for example, of these dragonfly wings. Multilayer reflectors, as opposed to single-layer reflectors, are periodic nanostructures that are organized such that their despacing is within the range of visible light. So I took the license to quote something that was written by Andrew Parker. Multilayer reflectors consist of parallel layers of a transparent material separated by layers of a material of a different refractive index. Such a structure can act as a reflector when the optical thicknesses of all layers are comparable with the wavelength of the incident light. As the angle of incidence changes, different wavelengths constructively interfere, and this gives the phenomenon of iridescence. The most efficient reflector, well, so on and so forth. What interests us is a broadband, wavelength-independent reflectance appearing silver to the human eye can be achieved in multilayer stack with a relatively disordered arrangement of layer thicknesses, about a mean value, and this is the case of the reflectance of fish skin. So what are we dealing with? The parameters that are important here and that we'll need to determine if we want to characterize such a multilayer reflector are the layer thicknesses and their indices of refraction that together give the optical thickness of the layer. The number of layers, the angle of incidence, and of course, and the maximum reflectance is obtained in a so-called ideal quarter wavelength stack when the optical thicknesses of the two layers are about equal and are in the visible range. Now, in organisms, what kind of media do we have that give this multilayer reflectors? We have pairs of media such as chitin and air, and these are their indices of refraction. As chitin with the cytoplasm, what is inside the cells, or different organizations of chitin. And then there is the systems of guanine crystals and cytoplasm. And what is particularly important is this number. You see how high it is, so how large is the difference between the two indices of refraction, and this is what we'll talk about quite extensively. So guanine crystal cytoplasm systems are found in fish and in spider and can give rise to silver, white, or iridescent systems. And we'll start with the fish scales. Now, this is a highly reflecting fish scale, and the reflectance is due to a number of cells, many cells, that are aligned under the scale, and each one of the cells is full with 10 to 12 very thin crystals. And the lines that you see here are in fact the crystals. So that light that comes from perpendicular to the scale will go through some 2 or 3 cells, and the light is reflected by the crystals. And the component of these crystals is guanine. And in fact, guanine had been known to be the component of the crystal for quite some time, although surprisingly, the structure was not known. The structure that was known was the structure of guanine monohydrate that is held together by this very strong stacking interactions between the guanine crystals. But what is the real structure that makes up the crystals in fish became clear only a very short time ago. And in fact, it was after we had started to try to look at these structures that anhydrous guanine structure became known. Again, there are very strong hydrogen bonds keeping together the molecules in the plane, and very strong stacking interactions that keep the molecule together in the direction perpendicular to the planes, or more or less perpendicular to the planes. So the first thing that we had to do is to identify the structure, and in fact the structure corresponds to the one that would be calculated in a powder diagram from the structure of anhydrous guanine. But look what happens, and that's quite interesting. In fact, although this reflection is the dominant one, in the fish, this reflection is so much more developed than all the others that it was clear that there is a phenomenon of orientation. And in fact, it's so strong that even the higher order reflections that should not be developed, judging by the calculated structure, come up very, very strong. This means that the crystals that are very thin plates are probably delimited, develop this face as a very large face. And in fact, the pole figure confirmed that we are dealing probably with crystals that have as their main face this 102 face. And this was then confirmed by electron diffraction, and Avital Levin-Lior finally solved and reconstructed the morphology of those crystals, and we now know what it is. Why am I going through that? Why is it interesting? It is interesting because it turns out that the index of refraction of the crystals in the direction where they are hit by a light in the fish or in the perpendicular direction is very, very different. And it is only in this direction that there is this extremely high index of refraction that contributes to the capability of these crystals to form a photonic system in the fish skin. And it is interesting also because if one calculates, as we did, the theoretical morphology, the morphology that we would expect these crystals to assume in a vacuum or in an appropriate environment, is a prismatic morphology where the direction that is developed as a main face, the face that is developed as a main face in the fish is in fact the less developed. So this means that the organism goes through a lot of pain in order to force these crystals to form as very thin plates so that they can achieve a reflectance that is much higher than what would have otherwise been. This is just to show you that the theoretical morphology is not model dependent, that there are some small differences, but by and large, these crystals must be prisms. And then can we reproduce that? Well, actually, when Avital Lévy-Lior first succeeded in crystallizing these crystals, she did that from the MSO. And then we were kind of scratching our heads because they still were thin plates that developed a little bit thicker than the crystals in the fish, but they were thin plates. And it was only when we started to use water, that is in fact the medium where organisms make their crystals, that we did find the prismatic morphology. And as we go down with the percentage of DMSO in water, indeed the morphology that was predicted theoretically appears, and this is practically identical to the theoretical morphology. So the conclusion is that these crystals are designed to enhance reflectivity, and inhibition of the crystal growth in the direction that is desired produced thin plates that enhance the light reflectivity of each crystal and thus of the whole multi-layer array. But then we can ask, how is the reflectivity regulated? And can we make a simulation of what the reflectivity is? And in order to do that, we have first to get reliable parameters from the system. Now, this was, again, something that was unknown until a very short time ago. And the reason is that what the scientists that studied this system had been able to do was to study them by transmission electron microscopy. And these are dried samples, and dried samples, of course, don't have the same parameters of despacings that we want to measure. How can we do that? We can do that by using cryoscanning electron microscopy that looks at the system as is practically, with no embedding, no drying, no other extraneous solvents or protectants, and looks at the system completely hydrated. So this is really the manner in which the cells are juxtaposed under the scale. And then we can see very well in these very thin slits, and we can see the crystals. Each crystal is enveloped in a membrane that's called a crystal chamber. And you can see one here that during freeze fracture was left over on this side, and you can in fact appreciate how thin they are. And there are, as I said before, about 8 to 12 crystals per cell. And now we can, in fact, get all the numbers that we want. Again, these are the thin crystals. So you can see the edge of the crystals here, and this is the cytoplasm, the cellular material, the cellular water-based material that separates between the crystals. And the measurements tell us that the average crystal thickness is 19 nanometers. So really, really very, very thin. And we can evaluate the average spacing between the crystals such that now we are well armed with all the data that we need in order to simulate and understand the reflectivity profile. And the reflectivity calculated for such a multiple periodic reflector that's built of n layers, and we know n, with refractive indices that we know because we have determined them, and thicknesses that we also now have measured. These are the simulated reflectivity. At the beginning, we weren't quite sure what we were looking at, but interestingly, introducing a nearest neighbor correlation of 60% that corresponds to the number of crystals that are better correlated because they are inside the same cell, we do obtain a very nice profile that tells us that about 50% reflectivity should be achieved with such a system, with a broad peak in the green-blue range that's appropriate to working in water, in the seawater, as the fish are, and that pretty well matches the measured reflectivity. So we think that we understand what goes on here. And now, at this point, I got a call from Jeff Oxford from the UK. And Jeff Oxford said, you know, I saw your paper, and it's very interesting because I'm working with spiders. And spiders, some of them come in a number of flavors. And there are white spiders and there are silver spiders. And they have guanine crystals in their skin. And the guanine crystals may be thin plates or maybe blocky prismatic crystal. So would you like to take a look at them? And I said, of course. And so started our adventure with spiders. Now, spiders form their crystals in cells that are called iridophores, again, and that line the intestine under the skin layer. We had, first of all, to determine whether the crystal structure of guanine that we were dealing with was the same. And we ran a series of powder diffraction pattern of silver-colored and matte white spiders. Now, there are two things that I want you to look at here. First of all, the silver-colored spider show the same effect of orientation as the fish skin did, with this very highly developed reflection that is a telltale sign of those thin plates, while the matte white are not oriented. And the second thing is that there is a lot of amorphous material, especially in the silver-colored, while in the fish there was nothing. In the fish it was all crystalline. So the crystals turn out to be, in fact, the same type of thin plates that develop in the same manner developing the same large face that we already saw is so useful for achieving high reflectance. And the Paul figure confirmed that, and the electron diffraction does. But look what happens here. All the crystals give the single crystals, or what looks like a single particle, give a double diffraction. And then one could say, okay, we know that that's certainly twinning. But no, it's not twinning because the angle is variable. We have all kinds of angles. So these are two crystals of amorphous guanine that for some strange reason always appear together in doublets. When we look at the cryo-SEM, now it looks much more messy. Here, I hope that you see the thin crystals as we saw in the fish. But in the fish, it was a fantastically, periodically ordered system. Here, it's not. We have all kinds of orientations of the crystals. But there is a short-range order. There is a certain persistence in the short range. And then, when we looked at the crystals at high magnification, they are indeed all doublets. They are doublets of crystals with a material in the middle that does not evaporate when we etch. So this is a solid material. This doesn't look like water. And then the question connecting to the amorphous material that we saw in the powder diffraction was, can it be amorphous guanine? This layer that is so visible in the middle, can it be a sandwich of two crystals with amorphous guanine in the middle? And in order to show that, in fact, it was quite easy, we dried the crystals, and when we dry the crystals, the thickness of the crystal goes down. And, but it doesn't go down as much as we would have expected if this was a layer of cytoplasm that's practically water. So it went down only of a few nanometers, much less than what we would have expected if it was water or cytoplasm. So we conclude that in fact, the layer between the crystal doublets is amorphous guanine. And now, again, we can calculate the whole thing. We can simulate the reflectivity. And we have the thickness of the crystal doublet. We have the distance between the doublets. We estimate, we don't have a precise measurement yet. We estimate that if this is a sandwich of guanine with crystalline and amorphous layer in between, then the index of refraction of the composite material should be around 1.7. And in fact, if we simulate the reflectivity based on cytoplasm in the middle between the crystal doublets, we obtain pretty lousy combination. But if we use the index of refraction of the amorphous guanine, the reflectivity is much higher. So taking into account the whole thing, we succeed, or the spiders succeed, in getting a percent of reflectivity of up to 40% that is uniform over the visible spectrum, and it is in all directions, because in fact, light in the spider comes from all possible directions and not as in the fish, only from one direction. So this explains why they are less ordered. And finally, let's take a look at the matte white spiders. You can see here these sacs of cells, of iridophores. And when we look into the iridophores, we see that there are cells that are completely chock full with crystals. And the crystals are not ordered, and they don't look like thin plates. In fact, when we isolate them, they are prismatic crystals. And in the cytoplasm, we can even see crystals embedded with very nice shapes. But there are also vesicles. And the vesicles are full of a material that is not etchable, so it is probably also amorphous guanine. And in fact, in some cases, we can see a crystal formed in the vesicle from the amorphous material. Now we have indeed what we were talking about and what we were expecting. So the matte white spiders, different from the silver spiders and from the fish, develop crystals in a morphology that is what we would have expected to obtain without any interference from the organism. In the matte white, the answer is quite simple. There is no crystal plates, no periodicity, no photonic arrangement. Light is just dispersed, scattered, and reflected at random. And this gives rise to the white color. So now to wrap it all up, we saw that both fish and spiders use the same material for the same function, but organize it in a very different way in order to optimize the reflectance of the crystals. The morphology structure relationship is controlled in such a manner that it enhances light reflectivity, resulting in reflectivity that compares well to the observed one. We saw inhibition of growth in the fastest growth direction, both in biogenic crystals and in vitro. And then the question is, it's the next question, how do they do that? I have no idea. The only idea that comes to mind looking at those crystals formed in the vesicle is that we are dealing with template enucleation of crystals from the appropriate crystal faces at the same time in vesicle that contain amorphous guanine. Whichever way it is, and this is something we're looking at right now, we conclude that fish and silver-colored spiders that are very far away in the evolutionary tree evolved efficient mechanism to control the morphology of the guanine crystal in order to optimize in slightly different manners the reflector properties of the skin, taking into account the environment in which they are, and it's all based on nanotechnology. What is left for me to do is only to thank the people who have contributed to this work. Of course, Steve Weiner, my colleague, Avital Levilior, Osip Schwartz, and Virgur are students who did the work. Danoron helped with the simulation. Jeff Oxford introduced us to the spiders. Berta Sivan helped us with the fish. And Fred Gavish also with the spiders and all the centers for electron microscopy. And thank you for your attention.